What's up, distinguished boys and girls? Welcome back to my channel. I'm reading straight from my PDF, so make sure you go and check it. I'll link it in the comments. We're gonna discuss the metabolism here. You should know that metabolism is divided into two processes. We have catabolism and anabolism. In catabolism, we will have degradation of materials, meaning breakdown of materials to produce energy. And in anabolism, we will have buildup of new compounds, and it requires energy. For example, in glucose particles, we can build it from CO2 plus H2O. This is usually in green plants, and it requires energy, as I said. Green plants obtain this energy from the sun. You should know that the glucose, or the sugar, is the main body's energy source. Okay, so our body's main energy source is the glucose. Because glucose can undergo a series of reactions to transform into pyruvic acid, and the process we call glycolysis. Now, glycolysis is a catabolic process, meaning a breakdown process. So in this process, I'm breaking down sugar into other molecules in order to obtain the energy from it. So we will start from the glucose molecule, the simple main source of energy. We will phosphorylate it into glucose 6-phosphate. Now, the word phosphorylation just means you add a phosphate group to it. So when we have a glucose molecule, we're just going to add a phosphate group to it by a process called phosphorylation. And we will obtain the glucose 6-phosphate. This process consumes 1 molecule of ATP. Remember that phosphorylation consumes energy. Now, the glucose 6-phosphate will transform into fructose 6-phosphate by means of specific enzyme. Now, fructose is also a sugar, but it's a different type of sugar. Fructose 6-phosphate will also get phosphorylated into fructose 1,6-bisphosphate. And remember, any phosphorylation we have will consume energy. So here we will consume another ATP. Fructose 1,6-bisphosphate will split into 2 glyceraldehyde 3-phosphate G3P molecules. Now, just to get that clear, this is not gonna happen directly. You have to understand that fructose 1,6-bisphosphate will initially split into 1 G3P molecule and 1 DHAP molecule. DHAP is the dihydroxyacetone phosphate. The DHAP then will convert to G3P, and it will yield 2 molecules of G3P. So initially, we have only 1 G3P and 1 DHAP. DHAP will convert into G3P, obtaining 2 G3P. I want to get that clear because you have to know this if they ask you about the diagram, because in the diagram they put the DHAP, but they do not mention it here. So I just want to get that clear. Now, each G3P will be converted to pyruvic acid, meaning we will obtain 2 pyruvic acid molecules. Because, as I said, we have 2 G3P, so each G3P will be converted to pyruvic acid, and the net will be 2 pyruvic acids. The conversion of pyruvic acid into the conversion of G3P of glyceraldehyde 3-phosphate into pyruvic acid will produce 4 ATP, 4 molecules of ATP. But you have to understand that the net ATP produced from glycolysis is only 2 ATP, because 2 are consumed in phosphorylation. So if they ask you about the net ATP, produced from glycolysis, it's 2 ATP, only 2. But if they ask you about the ATP that is obtained from the conversion of G3P into pyruvic acid, there are 4 ATP. And the reason is because we consumed 2 ATP in the phosphorylation process when we phosphorylated the glucose into glucose 6-phosphate, and when we converted the fructose 6-phosphate into the fructose 1,6-bisphosphate. If respiration is anaerobic, anaerobic means there's no oxygen, the pyruvic acid will go into fermentation. We have 2 fermentations, either alcoholic or lactic acid fermentation. And remember that fermentation takes place in the cytoplasm. Any fermentation takes place in the cytoplasm. But if the respiration is aerobic, meaning oxygen is present, the pyruvic acid will turn into acetyl coenzyme A. Acetyl coenzyme A will react in a cycle we call Krebs cycle in the cell's mitochondria. You have to understand that Krebs cycle, the aerobic respiration, is always in the mitochondria. But the fermentation, the anaerobic fermentation, which is alcoholic and lactic acid, is always in the cytoplasm. Now, if you see here, they mention 6C, 3C. Those just means the number of carbon atoms in the molecule. So 6C means the molecule has 6 carbon atoms. And 3C means the molecule has 3 carbon atoms. I want to clear something that is very vague in the book. When you see G6P, the glucose 6-phosphate, it means that the glucose molecule has a single phosphate group on a carbon number 6. Okay, so when you see glucose 6-phosphate, it means that I have glucose, and on my carbon number 6, I have a phosphate group. But the book referred to this molecule as glucose monophosphate, referring or indicating that there's only one phosphate group. But this nomenclature can actually be a bit misleading, as you might think the molecule mentioned is G1P, the glucose 1-phosphate, which is a completely different molecule. Because G1P has its phosphate group on carbon number 1. So I think when the book mentions a glucose monophosphate, I think this is misleading, because they should just mention it as a glucose 6-phosphate. But you should understand that the book indicating glucose monophosphate, they're just indicating that there is only one phosphate group in the molecule. Though this nomenclature can literally mean anything. They can mean the phosphate group can be in anywhere, maybe in carbon number 5, maybe in carbon number 4. So that's why I think this nomenclature needs some refining here, because it's not clear. The same case happens when the book mentions fructose diphosphate, referring to a fructose 1,6-bisphosphate, because you might think it is referring to a molecule like fructose 2-phosphate, which is wrong. Also, when the book mentions di instead of bis, this is also a wrong nomenclature, by the way. Because di is usually, the nomenclature, the prefix di is used when we have 2 phosphate moieties attached to each other. For example, when we use it in uridine diphosphate, UDP, or adenosine diphosphate, ADP. But in the case of 1,6-bisphosphate, we use bis because phosphate groups are not attached to each other, so they're on different atoms. So that's why the bis nomenclature is more correct and more accurate form. But the book mentions it as a diphosphate. Randomly, you can also encounter that biphosphate, but some authors consider this nomenclature as wrong or out of date. Now, the reason why I'm mentioning this is because examiners will simply just want you to memorize the false information straight from your book. So you can just unread all of that and just memorize the wrong information from your book, because that's what they want in the exam. Here I also put some the carbon atoms. Apparently they want to know it, so they want to know each molecule how many carbon atoms they have. So you should memorize them. Now, let's say that I've gone through glycolysis already. I've obtained my pyruvic acid from the glucose molecule. Now, what's going to happen to the pyruvic acid? If I have my environment in the cytoplasm with no oxygen, anaerobic, I will go through fermentation. I have either alcoholic fermentation or lactic acid fermentation. For the lactic acid fermentation, you have to know that, there was a question telling you to compare them together, which you should read very well. In alcoholic fermentation, it takes place in yeast, green plants, and some type of bacteria. First, the pyruvic acid is oxidized by taking the CO2 molecule, which also we can call it as a decarboxylation, but it's not mentioned in the book, so you don't have to mention it. The pyruvic acid then will be converted into acetaldehyde by oxidation. Acetaldehyde will be reduced by the hydrogen formed in glycolysis, and acetaldehyde will be converted to ethanol, which is an alcohol. Remember that the hydrogen here, the reducing enzyme here, is obtained from glycolysis, okay? And for lactic acid fermentation, simply the pyruvic acid will directly convert into lactic acid by just reducing it with hydrogen. It's very simple and direct process here. But let's say that I have my pyruvic acid in aerobic condition, I have my oxygen, and it is in the mitochondria, so I will go through the Krebs cycle. Krebs cycle is very simple. Just, by the way, this cycle is not complete in the book, so they just give you the most basic, the tip of the iceberg of Krebs cycle. So I'd be surprised if you think this is hard. It's very easy. Acetyl coenzyme A will be converted to oxaloacetic acid, and this will be converted to citric acid. Citric acid will be converted to alpha-ketogluteric acid, and the cycle will just keep going. You should memorize this as if they can tell you to draw the diagram of Krebs cycle. Now, you have to know that the whole energy, the ATP produced by Krebs cycle is 12, okay? And you should know that we have to go through Krebs cycle twice. So we have to repeat Krebs cycle 2 times in order to oxidize 1 gram molecule of sugar. And the energy released from oxidizing 1 gram molecule of glucose during aerobic respiration is 38 ATP in total. And this is how we get it. You can have a question tell you to prove that oxidizing 1 gram molecule of glucose sugar during aerobic respiration yields 38 ATP, and you will just write this down. We get 2 ATP energy gained from glycolysis. We said that the net is 2. And we get 3 ATP from the conversion of pyruvic acid into acetyl coenzyme A. We get 3 ATP, but we are going to multiply it by 2 because we are going through the Krebs cycle twice. That's why I'm going to multiply it by 2. So the net will be 6. We're also getting 3 ATP from the hydrogen resulting from the glycolysis after passing through the electron transport chain. Now, this point here, I just want you to memorize it because the electron transport chain is a very, very long topic that you don't need to know for now. But if you're really just that interested, you can look it up. But for the exam, you don't have to know anything about the transport chain reaction. And finally, we're getting 12 ATP from the Krebs cycle. And we're going to multiply it by 2 because, as I said, we are going through the Krebs cycle twice. So the total here will be 24 ATP. So 24 plus 6 plus 6 plus 2 will sum up to 38 ATP. And during anabolism, we will reduce CO2. One question you can have is justify or explain. CO2 reduction is considered a key process in the anabolism of the organic materials. The answer here will just be because plants, when they absorb the CO2 using water and solar energy, solar refers to the sun, by the way, to produce the carbohydrate materials. Now, carbohydrate means, again, CH, organic molecules. This reaction is called CO2 reduction. It leads to the formation of complex organic molecules, making it a key process in the anabolism of organic materials. Very simple. I don't see where the hard part is. Just 3 questions. You can have questions like, what is the chemical composition for each of the following, and they tell you pyruvic acid. You will write down the formula here. Also pay attention to where each process takes place. As I said, glycolysis takes place in the cytoplasm. Meanwhile, the glycolysis takes place in the cytoplasm, okay? As well as fermentations. But the Krebs cycle and the electron transport chain takes place in the mitochondria. Also, you can have a question. Krebs cycle produces 6-carbon acid called what? Now, if you remember from the diagram, we said it's citric acid. That's why you have to memorize the diagram very well, because you can have questions from them. Also memorize, like, where is the ATP produced, what is the hydrogen, the number of hydrogens. Okay. The comparison question, as you can see it here, compare the fate of hydrogen produced in glycolysis during alcoholic fermentation and lactic acid fermentation. You will just write down that in alcoholic fermentation, the acetaldehyde here reduced by hydrogen into ethanol alcohol. And in the lactic acid fermentation, it's a direct one. The pyruvic acid get reduced by the hydrogen to lactic acid. That's it for now. If you like the video, please subscribe to my channel. And thanks for watching.